Welcome to NST 222 Study Session 8 Normal Immune Response. Introduction. The human body naturally possesses the ability to identify and protect itself against bacteria, diseases, and substances that appear foreign and damaging. This is possible because of the presence of the immune response. It defends the body from possibly damaging substances by identifying and replying to antigens. Antigens are materials, usually proteins on the surface of cells, diseases, fungi, or microorganisms. Dead substances such as poisons, substances, drugs, and external particles such as splinter can also be antigens. Learning outcomes. After this study, you should be able to define immunity, explain anatomical structures or components of the system, highlight the properties of the immune response, identify the types of acquired specific immunity, and highlight the functions of the immune system. Meaning of immunity. Immunity can be defined as a state of responsiveness to foreign substances, which may be biological, microorganisms, chemical, drugs, etc., and tumor proteins. Put it differently, immunity is the ability of the body to recognize self from oneself, antigens, for three functions: defense, homeostasis, and surveillance. The picture below shows an image of an immune system and how they function in various parts of the body. See picture. Anatomical structures or components of the immune system. The immune system anatomy is complex. It consists of the following structures: physical barriers, the leukocytes, lymphoid organs, mononuclear phagocyte system. Physical barriers. Skin. The outermost layer of the skin is composed of keratinized, water-resistant squamous epithelium. Secretions of lactose that lowers the pH of the skin makes the skin more formidable and a less hospitable environment. Sweat from the skin contains salt, which toxic to pathogens. The sebum secreted by the sebaceous glands on the skin traps invaders and inhibits some forms of bacteria. An intact skin and mucous membrane prevents pathogens from gaining access into the body. The picture shows an image of human skin as one of the sense organs and a component of the immune system of the physical barriers. The lymphoid tissue associated with the skin consists primarily the lymphocytes and Langerhans, cells found in the epidermis of the skin. When Langerhans cells are depleted, the skin can neither initiate an immune response nor support a skin-localized delay hypersensitivity response. The respiratory system. The nasal hair and turbinate, in combination with mucous membranes, filter and trap pathogens from the upper respiratory tract. The secretions contain immunoglobulin A, IgA antibodies. In the bronchus, the mucus secreting cell and cilia bring the foreign particles to the carina, where the cough reflex is stimulated. This reflex helps to expel the invading substances. The epitheliums of the lungs secrete two proteins, surfactants A and D, which coat the pathogens, making them more visible for phagocytosis. The picture below shows the human respiratory system as process of breathing out carbon dioxide and breathing in oxygen to enable the body to perform its various functions. See picture. Digestive system. Beginning from the mouth, the saliva provides a chemical barrier. The pH of the saliva is slightly acidic and contains IgA antibodies, a bactericidal enzyme, lysozymes, that inhibits bacterial growth in the mouth and may help prevent tooth decay. The saliva also contains defensins, which functions as a cytokine to call defensive cells, lymphocytes, neutrophils, etc. into the mouth for battle. In the stomach, the presence of hydrochloric acid and pepsin is protective and invaders. Digestive system. Bile salts, fatty acids, lysolipids, and other digestive enzymes in the small intestine provide protection. In the large intestine are composed of a wide array of bacteria and fungal flora. This flora makes the intestines less hospitable to invading organisms. The picture shows the human digestive system, the process of intake of food, and proper digestion of the food substance in the body. See picture. The eyes. The lashes protect the eyes with blinking reflexes. Tears from the lacrimal glands contain antimicrobial properties such as IgA antibody, and also the tears wash out pathogens that bypass the eyelashes and the eyelid. The picture reflects the image of the eye as one of the components of human immune system, physical barriers, and the position of the eye in the body. How to protect the eye for proper sight. See picture. The vagina. In the vagina, lactobacilli, which constitute the normal flora of the vagina, secretes lactic acid that lowers the pH of the vagina, making it less hospitable to invading organisms. 2. The leukocytes. The white blood cells, leukocytes, are the main cells of the immune system. They are produced and stored in the bone marrow and develop from the stem cells. They spend most of their lifetime in the circulating in the blood. They are stored in the lymphoid tissues or dispersed throughout the host tissues. Lymphoid organs. This is composed of central and peripheral lymphoid organs. The central lymphoid organs are the thymus gland and bone marrow, while the peripheral lymphoid organs are the tonsils, gut, genital, bronchial, and skin-associated lymphoid tissues, lymph nodes, and spleen. Lymphocytes are produced in the bone marrow and eventually migrate to the peripheral organs. The thymus is directly associated with differentiation and maturation of T lymphocytes and therefore a significant part of the cell-mediated immune response. The thymus gland is enlarged in childhood but shrinks with age. With the aging process, the gland becomes merely a collection of reticular fibers, lymphocytes, and connective tissue. When the body is invaded by antigen, they move via the bloodstream or the lymphatic system to regional lymph nodes, where they interact with B and T lymphocytes and macrophages in the lymph node. The lymph nodes filter foreign materials, of which the spleen is the primary site for this function. In addition, the lymph nodes circulate lymphocytes throughout the system. It consists of two kinds of tissue: white pulp containing B and T lymphocytes and red pulp containing erythrocytes. Macrophages line the pulp and sinuses of the spleen. The spleen is the major site of immune responses to blood-borne antigens. If the spleen is removed in children, it can predispose them to life-threatening septicemia. Macrophages line the pulp and sinuses of the spleen. The spleen is the major site of immune responses to blood-borne antigens. If the spleen is removed in children, it can predispose them to life-threatening septicemia. Mononuclear phagocyte system. This system includes monocytes in the blood and macrophages found throughout the body and possess significant roles in immune response. They are responsible for capturing, processing, and presenting the antigen to the lymphocytes, thereby stimulating the humoral or cell-mediated immune response. Capturing is accomplished through phagocytosis. The macrophage-bound antigen, which is highly immunogenic, is presented to circulating T or B lymphocytes and thus triggers an immune response. Properties of the immune response. The immune system is composed of 5 important properties. These properties enhance its protective ability, thereby making it less harmful with a long-lasting effect. The following are properties of immune response. 1. Specificity. 2. Memory. 3. Self-recognition. 4. Self-limitation. 5. Specialization. 1. Specificity. When a foreign antigen, a foreign substance, enters the body, it stimulates a series of cellular changes which result in the formation of a specific antibody or sensitized lymphocyte to the specific antigen. 2. Memory. The immune system has the unique ability to remember a previous encounter with a specific antigen. Therefore, a secondary immune response is faster and stronger. 3. Self-recognition. This explains the ability to recognize body's own proteins and foreign proteins. 4. Self-limitation. A self-limitation response means that after the antigen is eliminated, the stimuli for the immune response is decreased, thereby decreasing and eventually eliminating the immune response. The self-limiting aspect of the immune response prevents damage to cells that would result from a prolonged response. 5. Specialization. The immune system reacts in different ways to various antigens and microorganisms. Types of acquired specific immunity. The following are types of acquired specific immunity: active immunity, passive immunity. Types of acquired specific immunity. See table. Types of immunity. There are two types of immunity. They are natural innate immunity and acquired immunity. The picture below shows the various types of immunity found in the human body and their active function. Adaptive immunity. Naturally acquired and artificially acquired. Active. Antigens enter the body naturally. Body induces antibodies and specialized lymphocytes. Passive. Antibodies pass from mother to fetus via placenta or to infant via the mother's milk. Artificially acquired. Active. Antigens are introduced in vaccines. Body produces antibodies and specialized lymphocytes. Passive. Preformed antibodies in immune serum are introduced by injection. Natural innate immunity. Innate immunity is the type that is present at birth, and it provides a first-line defense to pathogens. It is a non-specific response which not specific to a particular antigen. Acquired immunity. Acquired immunity. Acquired immunity occurs following an invasion of the body by microorganisms, resulting in the development of antibodies and sensitized lymphocytes. This type of immunity may occur actively or passively. Let us examine each of these forms of acquired immunity. Types of acquired immunity. The immunity of the human system can be divided into two. They are active acquired immunity, passive acquired immunity. 1. Active acquired immunity. This occurs when the body is invaded by microorganisms and antibodies with sensitized lymphocytes are developed. At a second exposure to similar microorganisms, there is rapid and more vigorous response to fight off the invader. Active acquired immunity may result naturally from a disease or artificially through inoculation of a less virulent antigen, e.g., immunization. Because antibodies are synthesized, immunity takes time to develop but is long-lasting. 2. Passive acquired immunity. This describes a situation where the host receives antibodies to an antigen rather than synthesizing them. This may take place naturally through the transfer of immunoglobulin across the placental membrane from mother to fetus. On the other hand, artificial passively acquired immunity occurs through injections with gamma globulins. Serum antibodies are given. This produces an immediate but short-lived effect because the host did not synthesize the antibodies and consequently does not retain memory cells for antigen. Antigens. An antigen is any molecule that can bind with a specific antibody. In other words, an antigen is a substance that elicits an immune response. The term immunogenic can also be used to interchangeably with antigen. Most antigens are composed of protein, although large-size polysaccharides, lipoproteins, and nucleic acids can also act as antigens. All of the body cells have antigens on their surfaces that are unique to that person and enable the body to recognize self. The immune system becomes tolerant to the body's own molecules and therefore is non-responsive to self. It is important that not all antigens elicit an immune response. Note, such antigens are called aptins. Therefore, aptins are low molecular weight substances that by themselves are harmless. But the aptins to elicit an immune response, it must be bound to a carrier molecule. This has a clinical significance because once the aptins are bound to their carrier molecule, the immune system will produce 3 different kinds of antibodies against them in this order. 1. The first antibody will react to the aptin regardless of whether it is bound or not. 2. The second type will react to the carrier molecule regardless of whether it is bound to the aptin or not. 3. The third kind of antibody reacts only to the aptin carrier complex. Common aptins include dust, animal danders, drugs, and industrial chemicals. Immune responses to aptins are the basis for many common allergies. Functions of the immune system. The functions of the immune system can be summarized in the table below. See table. The lymphocytes are the only leukocytes associated with the adaptive immune system. The lymphocytes proliferate, reproduce within the lymph tissue, and differentiate when activated into their final functioning form. This process allows cells that are able to respond to a specific invading organism to reproduce faster without a corresponding increase in the other lymphocytes, which are not needed for that specific trait. Functions of the immune system. In all the 3 roles of the immune system, the most important is the ability to distinguish between the body's own protein and foreign. There are a group of genes that are responsible for recognition of self or own protein. They are referred to as major histocompatibility complex MHC. There are 2 major types of MHC. 1. The class 1 MHC. The class 1 MHC proteins are present and known as human leukocyte antigens, HLAs. They are found on the surface of the cell membrane of almost all host cells. Any substance which lack HLA are identified as non-self. This means that each person has a unique HLA that identifies each individual. No 2 persons have identical HLA. However, identical twins may have similar HLA. The class 2 MHC molecules are found majorly in the cells of the immune system, but may be induced by other cells by interference. Interferons are proteins that are formed when cells are exposed to invaders, such as viruses, which are able to activate other components of the immune system. Humoral immunity. The humoral immunity is an adaptive immunity which consists of antibody-mediated immunity. The term humoral is used because all antibodies are produced by plasma cells, which is the body fluid. Humors, in Greek, arising from proliferation and differentiation of activated B lymphocytes. Production of antibodies immunoglobulins is an essential component in a humoral immune response. Immunoglobulins are composed of amino acids arranged on 2 light and 2 heavy polypeptide chains. Differences in the heavy chain configuration differentiate the 5 classes of immunoglobulin, which are IgG, IgA, IgM, IgD, and IgE. Each class of immunoglobulin has specific characteristics. Cell-mediated immunity. The cell-mediated immunity is that which activated through specific antigen recognition by T cells. Although the T cells are the primary cells involved in this reaction, other cell types such as T lymphocytes, macrophages, and natural killer cells are also involved. The importance of the cell-mediated immunity include 1. Immunity against pathogens that survive inside of cells, including viruses and some bacteria, e.g. Mycobacterium. 2. Fungal infections. 3. Rejection of transplanted tissues. 4. Contact hypersensitivity reactions. 5. Tumor immunity. For better understanding, let us describe briefly other cells which are involved in cell-mediated immunity. 1. T lymphocytes. This comprises of T cytotoxic, T helper, and T suppressor cells. T cytotoxic cells are involved in attacking antigens on the cell membrane of foreign pathogens and releasing cytolytic substances that destroy the pathogen. These cells have antigen specificity and are sensitized by exposure to the antigen. Just like the B lymphocytes, some sensitized T cells do not attack the antigen but remain as memory T cells. As in occurs in humoral immune response, a second exposure to the antigen will result in a more intense and rapid cell-mediated immune response. On the other hand, the T helper CD4 and T suppressor CD8 cells are involved in the regulation of the humoral antibody response and a cell-mediated immunity, being referred to as immunoregulatory cells. The number of T suppressor cells decrease in many autoimmune diseases in proportion to the number of T helper cells. This decrease results in an overaggressive immune response. For example, the human immunodeficiency virus, HIV, invades T helper cells, thus decreasing their number and function. Therefore, individuals with HIV infection do not mount an aggressive immune response and are at an increased risk for an opportunistic infection and malignancies. 2. Natural killer cells. These cells are not T or B cells, but are large lymphocytes with numerous granules in the cytoplasm and are often referred to as large granule lymphocytes. NK cells do not require prior sensitization for their generation. These cells are involved in recognition and killing of virus-infected cells, tumor cells, and transplanted grafts. The mechanism of recognition is not fully understood. NK cells have a significant role in immune surveillance for malignant cell changes. 3. Cytokines. The cytokines are small proteins which affect how cells behave. Each cytokine possesses its own kinase and kinase inhibitors, which are important in regulating immune responses. Kinase and kinase inhibitors are enzymes which destroy or preserve cytokine respectively. Therefore, too much kinase or too little kinase inhibitor will result in abbreviated immune response, while too little kinase or excess kinase inhibitor will result in prolonged immune response. Cytokines can be classified as lymphokines secreted by lymphocytes and monokines secreted by monocytes or macrophages. Cytokines instruct cells to alter their proliferation, differentiation, secretion, or activity. There are currently at least 60 different cytokines, and they can be classified into distinct categories. The interleukins, IL, and colony-stimulating factors act as immune modulatory and growth regulating factors for hematopoietic cells. The interference class of cytokines interfere with viral replication of previously unaffected cells. They are antiviral and immunomodulatory. 4. Macrophages. Cytokines attract and activate macrophages in the area of the immune reaction. These macrophages secrete cytokines that further modulate the immune response. In addition, they can release lysosomal enzymes that damage surrounding tissues. Antigen-antibody interaction. Antigen-antibody interactions result in elimination or destruction of the antigens. 5 specific forms of interactions are listed below. 1. Precipitation. Soluble antigens combine with antibodies to form a lattice of insoluble complexes that precipitate and are eventually eliminated. 2. Agglutination. Particulate antigens, e.g., red blood cells, RBCs, may combine with antibodies to form clones. 3. Opsonization. Bacteria are coated with molecules that allow them to be more easily recognized and ingested by neutrophils and monocytes. The opsonization mechanism involves IgG and complement-derived C3b. Opsonized particles attach to receptors on the surface of neutrophils and monocytes. For more information on opsonization, refer to lecture on inflammatory response. 4. Lysis. This occurs after the complement acts on the cell membrane of the antigen to cause rupture and leakage of cell content. 3. Neutralization. Antibodies neutralize some toxins released from bacteria. The mononuclear phagocyte system phagocytizes the antigen-antibody complex and removes it from the body. Comparison of humoral immunity and cell-mediated immunity. See table. Summary. In study session 8, you have learnt that 1. Immunity can be defined as a state of responsiveness to foreign substances, which may be biological, microorganisms, chemical, drugs, etc., and tumor proteins. 2. The immune system anatomy consists of physical barriers, the leukocytes, lymphoid organs, and the mononuclear phagocyte system. 3. Properties of the immune response are specificity, type 2, 3. Properties of the immune response are specificity, memory, self-recognition, self-limitation, and specialization. 4. There are two types of acquired specific immunity: active immunity and passive immunity. 5. An antigen is any molecule that can bind with a specific antibody. End of study section 8. Thanks for listening.